so to begin with, here are the materials. It is a 10-inch styrofoam ball, some 3-inch styrofoam balls and 1-inch styrofoam balls, then some acrylic paint, some craft foam, some thick white craft glue, a low temperature glue gun, and then of course toothpicks and white paper. Right, so then we have the necessary tools. And here we have some scissors, some measuring tape, a marker, a bar of soap, a paintbrush, a packet of rubber bands, and a teaspoon. Okay, so I started off with a cell by cutting the top half of the 10-inch styrofoam ball and repeating the process with the 3-inch one. I used rubber bands to divide up the ball into 4 equal parts and chose which top I should take through drawing guidelines with a pencil, as you can see here, and then cutting along those guidelines. So once I had completed the assembly of the cell, it was time to get creative. And so with my acrylics, I started to paint the main parts of the cell, beginning with the cell membrane, which I'm painting here. And the cell membrane was very, very important to paint first, because this is the semi-permeable membrane surrounding the cytoplasm of the cell. So in effect, it's basically the cell's hard protective shell. And as with that, its importance comes with that as well. So then I began to focus on the main inner parts of the cell, choosing red for the outer core of the nucleus and choosing pink for the inner core, finally finishing with blue for the nucleolus. Now, the cell nucleolus is actually specifically important because it contains the majority of the cell's genetic material in the form of DNA. And actual fun fact, each human cell contains roughly 2 meters of DNA. Now, the nucleolus is contained inside the nucleus and progresses the cell through its stages of interphase. So both of these components are actually very important inside the cell, since these two sort of interact as the main control centers of the cell. And what I basically did to create the nucleus and the nucleolus is I used the 3-inch styrofoam ball that was cut up for the nucleolus, and then I used the scrap of the 1-inch ball for the nucleolus.
Science!
moving forward, I went on to the next step and assembled the rest of the parts of the cell with my hot glue gun, sticking on the ribosomes around the endoplasmic reticulum, which I made out of folded up plasma foam paper, and covering that with covering the nucleus with the reticulum. Then I went on to put the mitochondria in place, and the reticulum is actually very important to the cell because it helps the cell become involved in protein and lipid synthesis as it goes through its different stages. And ribosomes are formed around the endoplasmic reticulum and are minute particles that consist of RNA.
She blinded me with science.
So finally, here's the finished model with all of the different parts of the cell attached and labelled. And apart from the parts that were explained at the beginning, there's still the Golgi body, which is the green mass next to the endoplasmic reticulum, which we see here, and it's responsible for the secretion and intercellular transport of the cell. And the reticulum and the Golgi body actually both interact together through the rough ribosomes of the reticulum that are able to produce proteins. And these proteins are then transported to the Golgi body, which sort of postmarks the protein to a specific location, such as the membrane. So yeah, hope you enjoyed. Thanks for listening.